My name is Darren Chooneea and my PhD was in microbiology. Specifically, my title was the characterization of the outer membrane proteome of Salmonella. My PhD project was, the grant was written by the Health Protection Agency. It's now known as Public Health England. And so my supervisors basically wrote the grant for the project, and just like when I was looking to apply for a PhD, this one came up. Read around the subject, it sounded pretty interesting, just how diverse the Salmonella species is. There's like over 2,500 different serotypes, but only one or two is able to cause disease in humans. It's only specific to humans, and Salmonella typhi, which I worked on, and it causes typhoid fever as well. So genomically they're very similar. So my project was basically to see what proteins are expressed that made them different, and more specifically just looking at the outer membrane proteins. When I started my PhD at the Health Protection Agency, they didn't have anything in place any methodologies in place to extract outer membrane proteins, and so I optimized a range of them. And also Salmonella typhi is a hazard group 3 organism. It can be used in a containment level 3 laboratory. It's as hazardous as people. It's handled in the same lab where people handle things like Yersinia pestis, which is the causative agent of the plague, and tuberculosis. So everything which I'd done could be transferred over to a containment level 3 lab. It can be, and people can follow on the project from there. So I basically arranged a range of methods that someone could work out, and I also had the opportunity to do some collaboration work with a group in Sweden, and the work we'd done there was like a novel method to characterize outer membrane proteins again. And that was a published paper in BMC microbiology. So yeah, and again, how that changes as well. Like, I can go on the BMC website and I can see my paper being cited in different journals. So slowly but surely, it's getting more and more impact, you can say. Because the Health Protection Agency was the affiliated research centre, I didn't really have much hands-on time with the OU per se. It was basically there to give me the degree. All my work was done, and all my support was all in-house, given to me by the Health Protection Agency. So, to be honest, it felt more like a job than an academic study, where me being in the university base, for example, because I also had a lot more difficulty arising, because you had to work around people whose jobs were there to work in the lab, so you had to work around them, other projects going on. So in the state of things, like a PhD student was quite low down the echelon, so you kind of had to sneak your samples on when you can and work weekends as well. But as I was told, like research is not a 9 to 5 anyway. You just have to get in, and when you get the time and opportunity, you do it. So.